Okay, yes, welcome, and thanks for coming. So, yeah, as Graham mentioned, it's not just about biofuel production from algae, but I also want to talk about our other plant biotech experiments. So really, the biofuel from algae is an experiment, and we have several of these experiments to look at food and energy security and see if we can address that. So, just need to acknowledge some of the, we work quite closely with industry partners as well, and of course with ARC and so on. So they're just mentioned here. And you're welcome to contact me anytime by my email, or we also have a web page, schenklab.com. So if I ask my students actually, and I can ask you as well today, what do you think are the biggest problems? What do you care about the most? So if I just ask you what is most important for you right now, what would you tell me? If someone's not. Pardon? Money is important? Yeah? Okay. Can I ask you a question? Can you eat money? Can you breathe money and can you drink money? No, right? So I guess it's not.
Do these things without money.
Yes, you can. Yeah, no, I've actually been in Papua, and I've been in the tribes there in the highlands, and they don't know money at all. But they certainly know that they need to get food and water, and they need to breathe air. So it actually comes down to food, water, and air. These are the most essential things that we all need. So the question is, where does that come from? Obviously, we cannot survive without food. So we need to look at food security. And in fact, human health is also important, of course. But the biggest disease is actually malnutrition in this world. So that's related to food security as well. So you can see it's really coming down to the food. Also, biodiversity decline, pollution, this has been mentioned as well. That's a big problem as well. We're losing some of the natural landscapes. And energy security, I'll talk about that as well, because that's very closely linked to food security. And then these other problems, obviously they exist as well. But they sort of linked, as far as I know, over the years from the students. They tell me this is sort of linked to these key problems. So we really should address these key problems. If we don't know what to do anymore as a researcher, I think it's good to look at these problems and see whether we can refocus on some of these. Because as a scientist, I think we are very privileged. We are trusted by most other people that we do the right thing and that we come up with brilliant new ideas. So really, if you look at it, we are privileged we're paid to play around and try new things. And I think that comes with a responsibility. So food and energy security are closely linked. Just wanted to, our population is going up like crazy. So basically, we live in a situation that is not the typical situation of the human population. The typical situation would be something that is more or less a stable population, like most animals would have. So, but currently, we're in this expansion phase. We're still in the expansion phase. Maybe it will level off eventually. But we are in an unusual situation of growth where the petri dish is still available, but we're seeing the end of the petri dish. So we need to be aware of that there is an end of the petri dish coming up, where we're growing in. And I just want to quote Richard Dawkins, who said, if there's ever a time of plenty, this very fact will automatically lead to an increase in population until the natural state of starvation and misery is restored. Now, that doesn't sound very nice. So there could be an alternative, and the alternative is that we are taking an active role in controlling what we're doing. So if we adopt sustainable practices, we should be able to stay around much longer. And don't go to a disaster and go into starvation and misery. Although starvation and misery, of course, is already a problem. We have 700 million people already suffering from malnutrition. And then we have this issue that food production must increase by 70% until 2050 using existing arable land. So that's a big issue because we cannot go so much higher with just efficiency. And as food prices go up because of there's so little land to use, we need to pay more for, the household needs to pay more, which is a big problem for developing countries, of course. So the unsustainable practices that we're faced with are, we have deforestation going on, we have erosion, soil loss, salinity as well. We have overfishing. I just show one graph here, which is global fish stocks. You can see how they're going down. Climate change is happening, oil dependency, groundwater depletion, peak phosphorus, pests and diseases, some say they are on the incline, and the meat demand increases as well. I looked at a recent publication in National Geographic, and you could see that nearly half the land is actually used to grow food for animals and not for us. So we could all turn into vegetarian, I guess, but then maybe half of coffee wouldn't be so happy, right? So maybe we... yeah, so, and then there's also the new pressure of biofuels and biomaterials. So if we go into a bio-based new economy, we actually have more pressure on the land to produce these compounds, biomaterials and biofuels. So it's still the same kind of land that we have. So, and you can see how the oil dependency is really showing that oil and food prices go really close together. You can see oil and food prices, they basically are the same graph. Because we use so much energy to produce food, it takes about 3 to 10 calories just to produce 1 calorie of food. So if you think about that, what you would eat today would be about the same amount as burning 1 liter of fossil fuel, just every day, just because you eat some food, because we use so much energy to produce it. So maybe we need to look at some, obviously we can do some plant breeding, maybe come up with resilient crops that will resist some of the stresses, and also sustainable practices. We need to move towards sustainable practices. So I want to talk energy security because it's so closely linked. So energy is a big problem because currently, if we look at the energy consumption, 80% of global energy demand comes from fuels. And if we look at the renewables, all the renewables are actually producing electricity. So we're missing out on 80% of the current energy demand if we just look at the renewables. So even if all small cars become electric cars, we still have larger trucks, we have boats, we have planes that still depend on fuel to be run. So we also have an increased attention on climate change, decline in fossil fuel reserves, increasing cost, environmental damage. So in other words, there is a big need for sustainable biofuel production, but without competing for arable land or biodiversity. Very important. We can't just destroy what's there, so we can't compete with food or cut down the rainforest. So the first generation biofuels unfortunately did exactly that. You can see here palm oil plantations cutting into the rainforest in Borneo. So that's a very sad picture. Or you have the situation that food is being used for fuel. And also not the right message, I think. So competition with arable land and food production is happening. And destruction of valuable natural areas such as rainforests is also unfortunately happening. So in our team, we just thought about, and this is really, I'm just talking about my students and staff here because they have most of the ideas. So they come up with some approaches. These are just some of the approaches that maybe we can go forward to address food and energy security. And there are many more examples, of course, but it's just something that we have picked that we might be able to contribute towards too. But of course, it's just a small contributions, and there are many hurdles. So I'll point out some of these hurdles as well. So one is we would like to have a molecular breeding of resistant or resilient crop cultivars, we call them. So this would be drought-tolerant cultivars that can cope with the variation in climate, for example. So this is more the traditional area that I've been working in. Then we're looking at new farming practices with beneficial soil microbes. So beneficial soil microbes, although they sort of exist and people know they do something good maybe, it's not really so clear how that works and if we can do something about it. Can we actually breed plants that live in better harmony with the soil microbes? Because the soil microbes play a big role, as I will show you in a moment. And then I'll talk about algae farms, because algae have the potential that they don't need to grow on arable land. And so they could be used for food production, for feed, and fuel. And if you're having one of those crackers there, you have already been falling into the traps of eating algae as food, because they're algae crackers. So I hope you like them. Anyway, so just looking at the first option, molecular breeding of resilient crop cultivars. So here, I just want to take a little bit of a step in plant physiology for you. So, I mean, most of you would know this, but plants obviously need light and water to grow. So if one of them is missing, it's a problem. Water is the biggest problem, really, because without water, the plants will not grow and cannot do their transport. So that's why the pathways required for abiotic stress signaling are the most dominant pathways in the plant. So abscisic acid is the hormone that usually upregulated during abiotic stress. And so the abscisic acid pathway is stronger than any of the other pathways. But there's also plant defense pathways against pathogens. And these are the salicylic acid pathway and the jasmonic acid pathway. Then there's different types of interaction with microbes, so you can have diseases. So if the pathogen attacks the plant, then there's two ways to deal with it if you think about what the plant needs to do. There are two types of pathogens. They're the types of pathogens that would kill the cells first, and they produce toxins and enzymes to degrade the cell walls. And once the leaves have become really soft and they're dead, then they eat of them. These are the necrotrophic pathogens, so they love dead tissue. And then there's the biotrophic pathogens, such as rusts, for example. Their trick is to make the plant believe they don't exist. So they infect the plant, and the plant doesn't even notice. They feed off the living structure of the living cells. So they're very different pathogens, and now it's up to the plant to actually recognize the right kind of pathogen. If they are dealing with a biotrophic pathogen, they need to make sure they recognize it. And then one very good strategy against the biotrophic pathogen is to sacrifice the infected cell. So that's why you get little lesions on leaves. And so that involves the salicylic acid pathway and programmed cell death and reactive oxygen species and so on. But think about if it's a necrotrophic pathogen, that would be exactly the wrong thing to do, because then the plant would be actively killing some cells, and necrotrophic pathogens love dead cells. So in that case, the jasmonic acid pathway must be upregulated, which counteracts reactive oxygen species production and counteracts programmed cell death. So basically keeps the cells alive, but then has many other defense pathways. And the same pathway, jasmonic acid pathway, is also responsible to interact with beneficial rhizosphere microbes. So it's interesting that the same pathway does both of these things. And then that can lead to induced systemic resistance. So it's a type of immune system, and basically there's a memory, the plant has a memory. Same thing when the pathogens infect. Maybe they infect this leaf here, but then subsequent leaves could be resistant because of systemic acquired resistance. So there's one problem though, because plants can't do everything at the same time. So if they are exposed to multiple stresses, they need to make a decision. I already mentioned that when when abiotic stress is dominant, that they have to look into that. And so we discovered a few years ago a key gene, or master gene, if you like, MYC2. And this transcription factor is a positive regulator for abscisic acid, for the abscisic acid pathway, but it's a negative regulator for the jasmonic acid pathway. So we didn't know why that was the case, but then we thought about it a little bit more, because when plants grow and they have abiotic stress, such as drought problems, they need to upregulate the ABA pathway. And so in that case, MYC2 would be upregulated. But then they may not have a problem with fungi that would attack, because they would be mostly occurring under humid conditions. So that's why in the plants, it appears that they are wired to make a decision either between putting all the resources into abiotic stress or against pathogens, against necrotrophic pathogens. But that also explains why plants under drought stress are more susceptible to pathogens, because they can't do both. So here we looked at just a few, the idea was actually to use regulatory proteins such as MYC2 to manipulate the plant defense pathway, the natural pathways, so we can create resistant plants or more resilient plants. So here's one of the ideas. The mediator complex is interesting. It's got about 20 to 30 subunits, and it's linking the transcription factor with RNA polymerase II. Have you ever wondered why there's transcription factor motif so much upstream of the promoter of the Tata box? It's because there's a mediator complex in between the transcription factor and RNA polymerase II, and they link the two together. And then, so basically these subunits, each one of them is a level of regulation of transcription. So we can, by modifying the mediator complex subunits, we can also change the way plants behave and respond. So for example, if we knock out one of those subunits mid-25, you can see that suddenly plants become resistant against this fungal pathogen, Fusarium oxysporum, which infects the roots normally. So that's how it normally looks like, but this one is resistant. Still gets infected, but it's resistant. However, the same plants get infected by Alternaria really badly. So it turns out that mid-25 is required for jasmonic acid signaling. In this case, the plant defense against this necrotroph Alternaria are broken down, and Alternaria can suddenly infect, which it couldn't do before. But in Fusarium, it seems to be a story where the plant overreacts to the infections, like inflammation almost. So if the plant doesn't react and just tolerates Fusarium, it seems to be better off than doing something against it. And part of the reason is also because Fusarium induces the wrong pathway. So this is what a lot of pathogens that we actually see that are successful. They upregulating the wrong pathway. So maybe a necrotrophic pathogen would upregulate the SA pathway and the other way around for a biotrophic pathogen. So they're tricking the plant into believing there's something else. Here we have two other examples, Met18, Met20A. They're also resistant, but you can see the phenotype of the plant changes. So there's multiple pleiotropic effect. So here's another example, and it's nice to see one of my former students, Gerard, in the audience, who did this work. So this lectin is a protein which is secreted by the plant cells into the cell wall. And so it wasn't quite clear why it's secreted in such large numbers. But as it turns out, when we purify the protein and add bacteria to it, it clumps up the bacteria. So it appears to be almost like an antibody that the plant produces. Of course, plants don't have antibodies, but it seems to act like an antibody. So it's secreted into the cell wall, clumps up bacteria, and if you overexpress this gene in a transgenic plant, the wild type normally gets infected like crazy with Pseudomonas, but the lectin overexpressing plants, they seem to show some resistance. So it looks like it can immobilize the bacteria at the cell wall level already. The same thing works with nematodes, although not as strongly. And we have no idea why that is still. So this is also Gerald's work. I don't think we know really why it happens. Maybe it's something to do with the gut lining of the nematodes. But there seems to be some resistance as well against nematodes. Something else I want to show, and from Shazi, who also sits in the audience here. So a few years ago, she had this idea that she wanted to look at microRNA from viruses. It's been known for animal viruses that they produce microRNA. So the idea was to look at plant viruses. And so she predicted microRNA precursors, and then she also looked at the way which kind of genes would be involved. And this is just a summary of the pathway of the model that she came up with. So it involves the microRNA processing machinery of the plant as well. So if the and then out of this idea, we actually filed a patent because, OK, maybe if the microRNA from the plant, from the viruses, target plant genes, and then these plant genes might be involved in defense against the viruses, maybe we can counteract that idea. So here we have the plant virus genome producing microRNAs, and these microRNAs would normally bind to a host defense gene that acts against the viruses. Now, by interfering with that binding, by simply introducing a point mutation or a few point mutations in the target binding region of the target gene, we can prevent the microRNA from binding. Hence, the expression of this defense gene against the viruses is active. Another way is to have a decoy sequence or a sponge to just capture all these microRNAs. And another one is just to look at a breeding population, see where there's natural variation in that region of binding, and then maybe you have a resistance gene against the virus. So this has led to a startup company, Nexgen Plants, actually. So we're working in this area. And although I can't tell you exactly what the microRNAs and the transcription factors are, but I can tell you that many transcription factors and kinases in the plant are targeted by viral microRNAs. And they seem to be involved in the plant defense mechanism. So again, if we can interfere with that mechanism and then we can restore the expression of those defense genes, we should be able to make plants resistant against virus attack. And so here we have done this. I won't go into the details, but basically, if we restore the GFP expression, we know that the target is no longer targeted by the microRNA because we introduced a mutation. So this is the mutated target, so it's now showing GFP expression again. So this is an indication for us that we can make plants resistant against viruses using this approach. And then, of course, with all these things, we have the problem that, oh, they are transgenic plants. Sorry, there's the GM issue. So you can't really do this. You can't really save the world because they're transgenic. So what can we do there? So because of that, we looked at some non-GM approaches. So I guess this is called an intragenic approach. So here, this is Katja's work. So we're looking at native regulatory elements. So we have the idea is to transform tomato plants, but just using tomato sequences. So we use a tomato promoter. We use a tomato terminator. We use one of the defense genes against the viruses. We express it just in a different way. Then we also need to look at marker genes, or maybe we can look at some marker genes, and there's a few marker genes that Katja is trying in the moment, and as selectable marker genes. And then what about Agrobacterium? So Agrobacterium has the left and the right border, right? So there's some extra sequences being introduced. it turns out that these sequences are also present in the tomato genome. So we are able to piece together a binary vector with the tDNA just made out of tomato sequences. So it depends whether that's going to be regarded as non-GM in the future, but hopefully it will be, because it's just tomato stuff into tomato. Then I had some students who were not so good and not so reliable, and so they were going to make transgenic plants, Arabidopsis plants, and make them resistant against the fungus. And then Matthew Timmons, he forgot to water them, and then eventually he watered them again, but they were already dead. They looked like this pretty much, or they looked really wilted. But then it turns out that the transgenic overexpressing ones had a recovery phenotype, so they were drought tolerant in a way. So that was kind of nice. And so that's just a little discovery, how we didn't expect that. So I wanted to go to the next approach, beneficial soil microbes. So beneficial soil microbes are so important. I don't think people realize how important they are. If you grow plants without any microbes under exenic conditions, they don't grow very well. If you add microbes to the same kind of conditions, you can see they get about twice the biomass. So now you can ask, okay, this is a strong effect, a yield effect. So something that agriculture should probably look at if such an important yield effect can be achieved just by adding microbes. So question is, what is the plant doing? So you can look at functional genomics, transcriptomics, maybe some gene expression in the plant. So we've done a microarray, and we've seen that a lot of the genes are related to nutrient uptakes that are involved with the microbial interactions. You can then also ask, what are the microbes? What kind of microbes are there? And so you can take this new approach because of next generation sequencing. You can do a metagenomics approach. You just isolate the DNA and sequence the DNA and find out what kind of microbes are there. However, that doesn't tell you necessarily what they're doing. So if you wanted to know that, you need to extract RNA, and in particularly messenger RNA. And then you can do metatranscriptomics. That's still a little bit tricky, but we're trying to do all of these things. So the rhizosphere of plants, obviously you have many microbes living in association with the rhizosphere. Many of you have maybe seen the tree of life, but if not, I just want to point out you are actually here, together with all animals, worms, and insects. And then next to it are all visible plants here. These are all the plants. And then next to it again is all fungi. So pretty much the visible world that we live in is just represented in these tiny 3 little branches. So the rest of it is microscopic. And this is just the stuff that we know. Only about 95% is currently probably unculturable, so we don't even know what it is or how it looks like. So this means we are surrounded by a microbial world. We live in a microbial world, and I think most of you are aware that you're outnumbered by 10-fold in terms of cells that live on you by microbes. So if an alien spaceship would abduct you and sequence you, they would only find 10% of human cells or human genomes in you. So a similar story probably with plants. Plants have multiple interactions also in the rhizosphere with microbes, and I won't go into the details, but basically there are good ones, there are bad ones, and there are ugly ones. Right? So the good ones would be promoting growth or maybe defend the plants against pathogens, and then the bad ones are pathogens, and the ugly ones are the pathogens that pretend to be good ones, but they actually are tricking the plant into pretending they're good, and then they are bad. So maybe they start good and then become bad. Anyway, just coming back to this idea, so the beneficial rhizosphere microbes. So let's have a look if we can look at find something about these. So the idea is, let's have a look at exogenous treatment of plants to manipulate soil microbial communities. Can we actually actively manipulate the microbial community in the rhizosphere? Can we do something about it? And can we harness this to make plants grow better? I think this is a new approach that people haven't looked at yet. So when we treat plants with jasmonic acid, just spraying the hormone onto the plant, we notice that the microbial community in the rhizosphere changes. So this is just a, you can see that there, oops, sorry. And then you can see that some of the microbes become more abundant after the jasmonic acid spraying than before. And it turns out that the ones that become more abundant are the ones that will bring some antimicrobial compounds or insecticidal compounds with them. So for example, Bacillus thuringiensis or relatives of Bacillus thuringiensis would be attracted. So it came down to this kind of model. So under normal situations, plants would support microbes that would help them grow better, so nutrients. But then under stress conditions, and this is what we're mimicking when we're spraying with jasmonic acid, they're crying for help. And they're getting the help in form of microbes that would then defend the plants, either against insects or against pathogens. So it's kind of an interesting idea. So we want to take this idea further, but we have not really fully understood how we can fine-tune this recruitment of microbes. But it's something we'd like to do. One thing we also looked at, what happens to the actual genes that are expressed in there, and we can see clear differences in the pattern of genes that are being expressed. The problem with that is there are so many different genes, so many sequences that we find that have no homology at all, that it's very difficult for us to actually say what the function is of these genes. But these are some of the genes that did have homology, that were upregulated, and I won't mention all of them, but they actually summarized here. Lilia summarized them. It turns out a lot of them are involved in virulence, stress responses, horizontal DNA transfer, which is interesting, antimicrobial compound production, and microbial competition, such as CD4 production. We developed also a microarray. This is a Metatranscriptomics environmental functional gene array. So this array is just printed with transcripts for microbes, and it can be used to find out what's going on with the microbes in a particular soil. So for example, here we have a sugarcane field, just different sugarcane fields probed with this, and you can see there's not much difference in the microbic gene expression. However, if we have a history of soybean cultivation on the sugarcane field, we can see that somehow the genes seem to be differing. So then there's also the idea to culture microbes, and maybe we can extract some compounds from the soil microbes directly. Although only maybe 1% of the soil microbes can be cultured, but if you have thousands of different species in the soil, that's still an interesting approach. So here's some results from Ahmad, where you found some in vitro inhibition of Phytophthora. And so that was interesting for us to look at. And then when he looked a little bit further, together with IMB people here in this building, he was able to do NMR and identify the compounds. So this is a diketopiperazine compounds. And so this is a compound active against Phytophthora, and maybe the first compound that has been found against Phytophthora from soil microbes. So there's also the idea that maybe we can use antimicrobial peptides that are already known and express them in plants. So here are some of the known ones from known antimicrobial compounds. So maybe we can express these in plants. Of course, we have to make sure they're not toxic to us. And here's another approach. We can not know at all what the kind of genes are doing from the microbes, and still see if we can find antimicrobial compounds. So here's a vector that would express any gene in any form. So it's got a codon slippage area, and which means that you can put in a gene in any reading frame. Because the problem with bacteria is they do transcription and translation at the same time. So whenever you isolate RNA, only a fraction of the gene transcript will be available. So you need to express it there. And then the idea is to express it in high throughput systems, and then look at the peptides and see whether you can do antimicrobial assays on it. So another frontier that we still have to get familiar with is the eukaryotic metagenomics and metatranscriptomics. We only looked at bacteria so far, but of course there's many eukaryotes, such as fungi and protists, in the soil. So we should look at those as well. It's just that their genomes are so big that they're very difficult to deal with in the moment. Of course, there's many fungi. But one thing we have tried is ecological genomics of microalgae, which also leads me on to my next thing. And you can see Katja here isolating a few algae and characterizing these. So you can now do 18S ribosomal DNA profiling, and you can find out what kind of microbes, what kind of eukaryotes are present. So we just looked at 3 different water samples here: Brisbane River, Wellington Point, and Mooloolaba. And you can see the eukaryotic microbial population varies quite a bit. These are just different replicates. You can see it's actually reproducible to some extent. So leads me to the next point. Algae are interesting because they can be farmed for food, feed, and fuel. And so they're an attractive, highly attractive feedstock. As I mentioned before, we need to look at something that's highly productive and that can be grown on non-arable land. So microalgae are 10 to 20 times higher in terms of area productivity than any other crop. That's at least in theory the case, because they double, sometimes they triple in one day. So you imagine you had a plant which would grow from this to this in one day. That's what algae can do. but only under optimal conditions. So we need to understand how we can keep them in this optimal algae bloom conditions. So then they can use any water source virtually, because they can grow in salt water, brackish water, even polluted water. So that's a good thing. And you have all year round harvesting, unlike many of the renewables. And there's many valuable byproducts, for example, protein-rich biomass, omega-3, all these kind of things. So here's a process. We collect some algae from rock pools. Rock pools are fantastic environments because there are very harsh conditions there. They might dry out at some point, and they get a lot of sun. Maybe they get very hot. So these kind of algae that are able to survive under these conditions have usually the capacity to accumulate a lot of oil, a lot of lipids in their cells. So you can see here, if they accumulate lipid bodies, it's a survival mechanism for them. So whenever they run out of nutrients, they still can do photosynthesis. They just stop dividing, but they can still do photosynthesis with the light, because they only need CO2 and water for that. And so they just use that energy from the sun and convert it to chemical energy and store lipids, which is a survival mechanism. So we want to look at these things because we're interested in the oil, because you can use that for biodiesel production, for example. So we also would need to look at how we can harvest those cells, because they are microscopic, tiny cells. You can't just scoop them out with a net. You have to find ways to harvest them. Traditionally, use centrifugation to do that. But centrifuges is not something that you want to use here, because that cannot be done in very large scale. And it also would be very costly to do that. So it would be very expensive to do. So we looked at algae that would naturally settle when you stop agitating them. So you mix them around when they grow, and when you stop, then they will just sink. So that's the theory anyway. So, and then there's also induced settling. So maybe they grow normally, and then you expose them to something and they settle. So I'll show you an example of that. Then we need to look at whether they grow well under outdoor conditions. It's good when they grow in the lab, but they must also grow in larger scale in outdoor conditions. So this is a very applied project. And so we look at the bioreactor on one of the buildings of the roof, on the roofs here, and then also open ponds. And then we need to look at extraction, harvesting and extraction. Here you can see some oil extraction. So these yellow parts are the lipid bodies. And so the idea is to extract the oil first, and then you can use the different products from the algae for different purposes. Oil can be used for biofuel, but then the remaining biomass, which is high in protein, can be used for animal feed or for us, for food. You can also digest it and produce biogas from it. And then also you should look at some high value products such as omega-3 or carotenoids, which can also be used. Now if you look at it from a commercial perspective, usually that's the best thing to grow for. But omega-3 and carotenoids, at some point in time, they will be saturated. If everyone else and his dog grow algae, then at some point in time, these markets will be saturated very quickly. But we all need to have food, and animals as well, and we need energies in the term of fuels. So these two markets are going to be there for a long term, and they cannot be saturated very easily. However, the problem is biofuel from algae is currently too expensive. Here's a comparison of pongamia, sugarcane, and microalgae. And although I don't agree the way this study was done, it's by far the most expensive is to grow microalgae, and mainly because of the harvesting, which here in this calculation included a use of centrifugation, which in our case we don't have to do because the algae will just sink to the ground by gravity. So there's potential also for yield increase and cost saving. So we're looking at selecting the best strains, of course. So we have hundreds of strains, and we collected those, and so we have a nice collection. And then we pick the fastest growing ones that produce the most oil. Then we need to look at efficient technology, but simple and cheap technology for installation, harvest, and extraction. So that's why we have produced these pilot plants. And then upscaling. So with the economy of scale, you'd hope that you also save some money. So you try and grow them on much larger scale. So we have a UQ demonstration plant, which is at Pinjarra Hills, and I'm going to show you some photos of that. We also need to link to other industry to maybe save some money. So if we use wastewater for them to clean up the wastewater, that would be a good thing, because then there would be some benefit for the wastewater industry. But then it would be not possible to use the algae as food. Okay, or maybe you can use it for CO2 fixation. The biorefinery approach is good because there you have multiple products and no waste. So algae strain selection, just a few examples here. This is one of our favorite strains. It's M8, comes from Maroochydore, Sunshine Coast. So a few lucky students went out there and collected this algae. It's got a little red eye spot, so it looks at you in the microscope. And then when it's under starvation, no more nutrients, it will produce a lot of oil and lipids. The whole body is full of it. And you can synchronize this as well so that all the algae would be full of the oil droplets. And then that's time to harvest them. And so you can harvest them ideally by settling, just auto-settling, so without any energy input except for gravity. So then we had to set this up in larger scale because impatiently we wanted to see whether it works on larger scale. So from the lab, you get maybe 20 liters the most that you can grow in the lab. And this has been set up on the Goddard roof, so that here's a bioreactor. You can see, though, it was not made to be really very cheap because this structure, for example, costs a lot of money. And so, but it was just to see whether we can grow them under outdoor conditions. So we built this one first. And then here you can see pictures from the roof of the Goddard building. And you can have the bioreactor here, which is just the bags. And then you have also the open ponds, the raceway ponds. So these just circulate the algae around open pond. So it's a bioreactor and open pond system. So ideally, we'd have a two-stage system as well, where we have the algae growing in exponential phase here with optimum nutrient supply all the time. Then we take half of them out into an open pond. The open pond, we add a little bit of water, but there's no nutrients. So we let them run out of nutrients. And then with the sunshine, within 48 hours, they produce lots of oil. And then we can harvest them, so we concentrate them. So this two-split system is a good way of avoiding contamination as well. So we looked at some other technology. How can we make mixing cheaper, for example? So here's a way we use an airlift. Most airlifts go vertically, but this is an airlift which takes into consideration the flow of the water. So the water goes down, and then on a diagonal line, it goes up again. So it takes the flow of the water into consideration, so and then it just basically, with bubbles, is able to push the water around. So it's using compressed air rather than a pedal wheel, which is the traditional way of pushing algae around. And then another student of mine, Kalpish, he came up with this brilliant idea in his PhD to use UV light. So here he has exposed algae to UVC light, which doesn't normally occur, but it stresses them so much that not only they produce oil under nutrient starvation conditions, but that oil doubles if you expose them to UV light. So we have doubled the yield of oil production by just simply exposing them to a little bit of UV light. Luckily, the same algae also have flagella, and the flagella drop as you add UV. So this means they can sink. They fall down after the UV exposure. So it's a nice way of doing lipid induction and settling at the same time. So now then, after this, we looked at the costs. How much does it cost to harvest algae? And these are just some of the more traditional ways. For example, centrifugation is the most expensive. Then there's also other approaches, for example, foam flotation, and we're using a Jameson cell as well, which is normally a mining instrument to get coal dust out of water, but we're using it to get algae out of the water. And then we use that sedimentation, which is just by gravity. Maybe you can add a flocculant, or you can add UV, so this is with or without UV. Some of them wouldn't settle unless you actually add the UV lamp. But you can see the cost has gone down quite a bit using that approach. So then we wanted to do this larger, so we looked at this Pinjarra Hills campus where there's 260 hectares, which are not being used very much. So we were able to set up a facility there. So we call it demonstration plant because at this point we wanted to see whether we can already do some economical production of microalgae. Although we had a few disasters such as soil erosion and so on, but basically we were able to get through that. And so this is the current state where we have 3 raceway ponds. So the way this algae farm is run is you have a starter which comes out of the lab originally. Then you have some pre-cultures that are grown under outdoor conditions. Then you have an inoculum pond. So the pre-cultures, so starter is about 20 liters, pre-culture 180 liters each. The inoculum is 4,000 liters, and the raceway pond is 40,000 liters. So you see, you need to slowly go up. You can't just jump and inoculate with one little vial from your algae. You can only inoculate with 10% as the minimum. And so we do that here. And also, it's important to have backup culture in case you have contamination or other problems there. And then we have the harvester here, which is just a V-shaped pond. So basically, the algae just sink to the ground, and that's where we can harvest them by settling. Then there's secondary dewatering, and then we have a solar dryer as well, which just has some hot pipes, and hot air goes through it. It goes up to 85 degrees in there, so quite hot. I should also say that the algae that we use, it's an MLA project, and if you've been to Dennis Poppy's presentation a few weeks ago, he mentioned this project as well. So MLA, we're actually producing microalgae for feed for cattle for the Northern Territory. So this means we are using Northern Territory strains, and they have some amazing abilities. For example, they can survive 45 degrees in a glass house, we noticed. And right now, as the water's only 7 degrees out there, they're still growing. So they have a really wide temperature tolerance. So it's quite nice to see. So yes, so the business model is that we have high-value products, but then once we extract high-value products, we can look at the biomass and biofuel, and we can cover these different things here. So what about oil extraction? So oil extraction is an issue because currently hexane is used for oil extraction. It's an organic solvent, which is not environmentally friendly, and also it's dangerous because it can explode. So another student, or two students, Ali and Furuk, are looking at oil extraction without using a solvent. So there's ways to do wet extraction. So Furuk has shown that you can do wet extraction. You don't need to dry the algae, which would save some costs. And there's also ways how you can explode the algae so the oil droplets simply come out. And then, but once they come out, one of the problems is that they don't necessarily are available because they may form an emulsion with the liquid, the water. So then the question is, how do you get the little oil droplets out of the water? So how can you break the emulsion and recover the oil from the emulsion? And then luckily Ali came up with the new idea where he had a filter. He invented a new filter, and basically you pour the water, the emulsion, through the filter, you can recover all the oil. So we're trying to optimize this process in the moment, hopefully it will work. But the principle of this is you have a filter here, we call it a hydrate gel filter, and then we pour the liquid on top of it, and anything that has a certain particle size or is hydrophobic will stay on top of the hydrate filter. Anything hydrophilic or very tiny will go through. So for example, chlorophyll goes through, but carotenoids don't, because carotenoids are hydrophobic, but chlorophyll is hydrophilic. So this is the kind of thing that we're looking at. So this technology, as it turns out, can be used also to concentrate algae and to get oil out of it. So that means it has a lot more applications. For example, if you have oil spills, you can use this to recover the oil from oil spills. Or you can concentrate juices with this. This is carrot juice, for example. Currently, there's no concentrate for carrot juice because whenever juices are concentrated, they're heated up, and that would make an ugly color for the carrot juice. So you can also remove microbes from water. So as you pour the liquid through, the microbes would be captured in the hydrate gel filter, and the water that comes out is without any microbes. So we tested that. We even included phages and viruses, and even those are captured. So now we have a water purification device, which we discovered more or less by accident because we wanted something to concentrate algae. So we're very happy with that, and we're looking for industry partners. And this is just some other high-value products. So for example, we look at omega-3, EPA. And so we looked at the best strains there that produce a lot of omega-3. And then we also look at omega-3 optimization. There's phytosterols as well, which some of them, they're in margarine. Phytosterols are supposed to lower your cholesterol. So there's also some algae that we found that are high in phytosterol. So this is Farok. He found this one algae which was really much, much higher. It's about, you can look at this, it's actually 2.5% of its dry weight is phytosterols, so it's quite nice. And there's some new plans for algae farms. So maybe at Gatton we're going to build one, but then one of the more immediate goals is to build some for farms in the Northern Territory. So this is in line with what Dennis Poppy was saying a few weeks ago. So we want to build farms for the cattle growers in the Northern Territory, because then they can have their own protein source during the dry season. Currently, soybean meal and cottonseed meal are imported for them, so they can feed the cattle. But they can, if they have their own protein source on the farm, that would be quite attractive for them. There's another idea just to use algae as an energy-only idea. So here we would look at a zero-input algae energy farm. So let's see if that's possible. We haven't really shown it yet, but we have some data. So if we just have sun, CO2, and water as the only inputs into the system, we grow some algae, then we concentrate the algae, we do wet extraction, we get the oil to produce biodiesel. The remaining biomass, which is about 80%, goes into an anaerobic digester. That will produce biogas. The anaerobic digester has some leachate, which is full of nutrients, so the nutrients can then be used to grow more algae. So we've already shown that that works in small scale. But basically, this system would allow you to produce biogas and biodiesel. So it's an energy farm that produces fuel. And it has virtually zero input. You don't have to add fertilizer, because fertilizer is being recycled. So I think that would be quite a nice thing if we can show that that works on large scale. And then we just work on the cost, how we can reduce this. There's also something I want to mention is Kalpesh came up with this idea to capture his own carbon from his car, because his GPS was actually telling him, oh, you emitted so much carbon dioxide. So he said, oh, what can I do? I work on algae. So he put this extension to his exhaust pipe, which captures part of his CO2. And then he was able to remove, I think, half of the CO2 emissions that he had on his car. And then he used that, basically that forms carbonate, and then he used that to grow more algae. Okay, so then you can think about maybe you could use those algae to go into this system, or you can use the algae to produce biochar and then bury them underground. If you did that, you have biological carbon capture and sequestration going on. So you're actually removing carbon from the air. So imagine you would drive your car, and by doing that, you're actually removing carbon from the air, right? So that would require maybe that the service stations would give you a discount when you deliver some of your cartridges when you capture the CO2. But anyway, that's just future stuff. So in summary, algae may address human health in terms of omega-3 and so on, biodiversity decline if we don't have to cut down rainforest, food security and energy security. So we're excited about this. We calculate we need about 10 grams a day to cover the most important deficiencies. And lastly, I just wanted to acknowledge everyone who's been working on this. And these are just the current people. And you can see some of them start very young. So quite amazing, really talented people around. So and what I've been talking about is really just their work and their ideas. So I'm just the presenter, really. So that's the microbe group. They also have their own website. This is the Nexgen plants team. And there's 4 or 5 postdocs on it, or nearly 5. Gaia is just about to submit her thesis, so she will also be soon part of this. And then this is the algae biotechnology team. And so, all right, please ask any questions. Thank you very much.